Hello, good morning, everyone, and welcome to the GAN program, Next Generation Sustainable Alternative Proteins for Food, Fundamentals and Technological Innovation. We are on the day 8 today. The first lecture is about lecture number 15, Emerging Innovations, Cultured Meat, Microalgae, and Insect Proteins. So, you all agree that meat is important. I have shown in this slide on the day 1 as well. So, we consume a lot of meat industry is one of the largest industries in food sector. Though we want to go for animal to plant, still meat consumption is not reducing. It's reducing, but not that fast or something like that. It's still a big business around the world. So if you look into this graph, you can clearly see that Americans and Europeans consume or historically been consuming more meat compared to Asians. So it's one of the largest food commodities. So it's very difficult to tell people don't eat meat, right? That's in their daily diet. They eat meat. For example, we also eat meat in for lunch and for dinner. And I can't say to my parents, okay, completely stop eating chicken. They are not happy, right? So it's very difficult to completely stop meat. But then I also discussed this slide showing that okay, we can make things in a very sustainable way. We can produce more meat in a small time. Maybe that will help the world, but that is not the case either. We have been trying to do many things to animals, and now people talk about animal welfare. So it's very difficult to change how we are producing our meat today. I'm not telling it's impossible, but it's very difficult. We have already reached the biological limit of most of the animals. They're producing highest amount of milk. Chicken is producing highest amount of meat within 38 days. So by eating small amount of food, they're trying to convert all they have eaten into protein somehow. We have tried everything, but maybe there is nothing left. So that's why we need to have some sort of transition. I can say that okay, just eat plant-based foods, but still people are no. My tongue doesn't eat like plants every day. I need to have meat at least twice a week or thrice a week. So we are in a dilemma at the moment. It's impossible to completely stop eating meat, right? The dilemma is like this: we need to have a transition from animal-based to plant-based in order to protect animals, right? There's an animal welfare. They say that, okay, I don't want to harm animals because animals also have the right to live like us. That's one of the dilemma. But still, I want to eat meat, right? Do you agree? That's a dilemma we all have, right? The second thing is that, oh, we also know that animal husbandry is responsible for highest greenhouse gas emission. So we need to save the environment as well, right? The third dilemma is that we want to save the environment, we want to have animal welfare, and we want to eat meat as well, right? So we are in a confused state, like what do we do now? So we need to have different strategies to avoid this dilemma. One of the strategies we discussed that, okay, you take animal based sources. In this I show insect alternatives, and then get into meat analog and then eat them, right? So now you're satisfying yourself, you're craving for meat, right? You took an alternative source for meat and you satisfied your craving for meat. But again, we talk about, oh, I don't want to eat insects because that's not in my culture. I don't like insect because probably they are harmful for my health. Consumer acceptance is going down, right? So now we have been thinking about, is there any other option? Then we thought, okay, we will start processing the food, right? We want to add so many ingredients and process it, and then we get I hope I have another figure. We get different structures and texture, all plant based here. That gives you a new taste and new texture. However, we still think this is not meat, right? It's still, we think it's not meat because oh, it's coming from plants, or how can I call it as meat? It does not have the same juiciness, despite we processed all this thing. It does not have the same juiciness. It does not have same fibrous thing we feel when you eat chicken or pork or whatever other animal. We still have problems. Now we have been thinking, okay, is there any alternative? And one of the alternatives is that Can we produce meat? Can we produce meat without animals? Can we produce meat without animals? We don't have any animals here, no living animals here, but still we are getting meat. And that is a new technology, not very new. That's a new technology, and that is called cultured meat. Am I clear? That's called cultured meat. Just to give you some rationale here, how are we producing meat today? So I need to draw a structure of a cow. This is my cow. Okay, it has a ear, huh? And this is eyes. It looks like elephant, right? No, sorry. This is my cow. Okay, let me make a tail. So now, how we are producing meat? We feed the cow, right? We feed grass to cow, or we feed rapeseed press cake to cow, right? What cow will do? Cow will nicely eat them, right? Now the grass or whatever fodder we are giving to the cow, it contains nitrogen, it contains amino acids, it contains carbohydrates, right? They will digest it, then they will resynthesize those nitrogen into new proteins, right? That's how the body works. So they resynthesize amino acid into new protein. They convert nitrogen, carbon, hydrogen into new proteins, and that new protein goes to milk, right? That is the milk we are drinking nowadays, animal-based milk, right? The second option is that the same protein is converting into protein after digestion and becoming muscle, right? That's the meat we are eating, right? So basically, we are converting a plant-based nitrogen into animal-based meat or milk. Do you agree with me? There's a lot of processing is involved inside the body, and that emits greenhouse gases, nitrogen oxides, methane, and all these problems we have already heard about, right? Now, we don't want to have this track, right? Because there is a life involved here. Do you agree? There is a life involved, and we need to kill the animal to get the meat at the end of the day. So we have a life involved, and I don't want to use this track. What will I do? I'll take a big plate. Okay, I'll take a big plate, and I'll take a small portion of tissue or a cell from an animal from some region. I'll explain that later. I'll put it on the plate. Okay, God has created organism in such a way that some will grow continuously. For example, let's take bacteria as an example, right? You take 2-3 bacterial cells and put it into so-called media. 2 bacteria becomes 2 million bacteria, right? Right? You take microalgae, 3-4 cells, you put it into a so-called media, and 3 microalgal cell becomes this much biomass within, let's say, 10 days, right? So they grow, and like that, animal cells also has a property, they can grow, right? Now, just to give an example, you have a bacteria, this is my bacteria. I said that they grow, one becomes 1 million by dividing themselves, they by reproducing themselves. Now, what do we need for this? Do they simply grow? We need a media. What do you mean by that media? Media contains
Nutrients.
nutrients. So somehow if you supply nutrients, in some cases you need to supply light, for example, microalgae. Sometimes you need to supply carbon dioxide for microalgae. Sometimes you need to supply oxygen for oxygen-loving bacteria. So if you supply everything they like, they grow. Same thing is true with animal cells. If you supply nutrients in the form of media, they proliferate, right? They divide, they differentiate, and they can become a biomass, right? Not the same as this meat, but equivalent. Not exactly the same, but equivalent. A meaty structure, a meat containing equivalent proteins can be produced, and that is called cultured meat. Is it clear? We are producing meat without using animals, but using animal cells, and that is called cultured meat. And let's do a little bit talk about history. This is not something very new. I was reading some history about this when apparently in 1930s, Winston Churchill, the British minister statesman, and he said that okay, we need to we need to find an alternative way to produce chicken, how to produce chicken drumsticks and wings without chicken. And then then please, this is something really new concept. Nobody has thought about eating meat cultured in a glass jar, because the meat were abundant outside and nobody thought about it. But because of this transition nowadays, people are keep thinking about, okay, we need to find an alternative way. In 2013, Professor Mark Post from, I think he's from Netherlands, if I'm not wrong, Maastricht University, I'm not very sure. And he launched the first cultured beef hamburger, the burger. Apparently, it costed $330,000. It's a huge sum. It's that much is costed simply because the technology was not easy. It was new. He had to try so many things, and then if he got a hamburger, and that costed so much money. Would you buy that one? Nobody would that buy that. But research went on, right? Research went on. They tried numerous other things to reduce the cost, right? They made the processing so easy. And in fact, in 2020, in Singapore, one of the restaurants sold the first meat analog, the first cultured meat burger, I think, if I was not wrong, in Singapore. So it has been marketed, it has been eaten, the government has given approval. Does that make sense? So it was nothing, nobody imagined back in the days that I would eat a meat produced in a glass jar or a bioreactor. But now in 2020s, it has become a reality. People are eating in different countries. Many countries has given approval, which I'll talk about that later. Does that make sense? It's a brief history. What I wanted to mention is that it was an actually impossible thing people would imagine, but it actually has happened because of this transition. It was super, super, super expensive. Now it's just expensive. It will become maybe cheaper in next 10 years or 15 years or 20 years. I have no idea. But this is the way forward. Hundreds of different companies are working on this one to get cultured meat. Now, I'll talk a little bit about the technology and how we are producing cultured meat and so on. But if you look into the literature, if you write about cultured meat, people use different terms, right? They are the same meaning, right? Cultured meat, cultivated meat, lab grown meat. But please note that usually companies are unhappy if they use the word lab grown meat, because it's more scientific, because it's not necessarily lab grown when it comes to supermarket. It is grown in a food industry, right? We don't call a lab grown soybean chunk. Do we say that one? No, but that's the reason why they don't want to use this term. In vitro meat, in vitro means produced outside, not in vivo, is ex vivo. Cell based meat, synthetic meat, also not a nice term to use, just to for avoid marketing term perhaps. And slaughter free meat, that is a good terminology because we are not killing the animals here, so no slaughtering. Craft meat, healthy meat, sham meat, artificial meat, this is also something industries would avoid using, because as soon as you use the word artificial meat or synthetic meat, consumers say, oh my god, it may be something plastic. So that's the reason they might not. But people use different terminology in scientific articles or in news media and so on. So, but just remember that we use the term cultured meat. I might use in my lectures two terminologies, often cultured meat or cultivated meat. They are the same. All right. Now, how we are producing cultured meat, we need to understand the basics here, how exactly they are doing. So in the next couple of slides, I'm going to teach you little bit basics. You need to have a little bit of cell and molecular biology background or biochemistry background, but not much is needed. However, I'm pretty sure you guys have studied cell biology in your high school, in your colleges. It's a very common subject have been taught, right? We all know that the body is made up of numerous cells, right? The body is made up of numerous cells, and numerous cells can become a tissue. Do you agree? We have muscle tissue, we have brain tissue, we have heart tissue, right? numerous cells associate to form tissue, right? And numerous tissue can associate to form an organ, right? For example, you have a heart, multiple tissues are there. You have brain, a fat tissue is there, a protein tissue is there, right? It's protein enriched tissue is there. It becomes an organ. So we have all these things associated to make a living being, a system, right? I'm telling you about the complexity we have: cells to tissue, tissue to organ, organ to system, right? And some things are edible, not human beings. Let's say pig as an example, or chicken as an example, poor chicken, right? Drumstick, the meat is edible. Liver is an organ that is edible. Heart is an organ that is edible. Skin is there, right? Not necessarily. So that is also edible. So we have different things. At the end of the day, they all constitute as a broad category as meat, and they become our food. Now, important thing here is the most fundamental thing here is the cell, right? We start with the cell and we get the tissue, right? That is the way we are producing meat. Now, I'm going to talk a little bit about animal cells and how meat originates from cell and what kind of cells are there in meat. Is it okay? If you look into a meat, the animal meat, chicken meat, or let's say beef or pork, so we have three types of meat: skeletal, three types of muscles: skeletal, smooth, and cardiac muscles. Skeletal muscle is basically the muscle, let's say leg piece and the real red meat, what you see, right? And that is skeletal muscle, right? Cardiac muscle is maybe heart muscle, and then we have a smooth muscle. Usually, it's a muscle type present in the inner layer of intestine or stomach, and the soft thing is called smooth muscle, and they are not really a abundant thing to eat. But the important thing we eat is skeletal muscle. Do you agree? The important thing we eat is called skeletal muscle. Now I'll go and show you a little bit about how the skeletal muscle look like, right? This is a chunk of meat. This is a chunk of meat, and this chunk of meat is composed of skeletal muscle. And the muscle is made up of muscle fibers. Muscle is made up of muscle fibers. Muscle fibers are actually a complex structure of protein, right? It's a complex structure of protein. Fibers contains protein. It's also associated with fat, some other cells and other things, mainly composed of protein. Let's say 90% of protein and 10% of fat and something like that. So it's also called as myofibrils, right? Muscle fibers are myofibrils. Now just look into this structure. It's a very beautiful structure. It clearly shows how the muscle fiber looks like in a meat, right? It composed of this muscle fiber, which is covered by this blue color sheet. Can you see? And that is called endomysium. These are connective tissue. They're covered by an endomysium, a smooth layer around them, right? And it's made up of collagen. You know, muscles contains collagen, right? It contains endomysium, and these muscle fibers are bundled together. Individual muscle fibers are separated by endomysium, and they are bundled together to form a structure called fascicle. Fascicle is nothing but a bundle of numerous muscle fibers, right? And these bundles are separated by another connective tissue called perimysium. Can you see the green thing here? Multiple bundle of muscle fibers are separated by a structure called perimysium, right? And everything is surrounded by epimysium. Is it clear? The muscle fiber is a complex structure. It is surrounded by a tissue called endomysium, and multiple muscle fibers that are bundled together, they called as fascicle, and fascicles are surrounded by perimysium. Basically, perimysium is the one that separates muscle fiber bundles, and the entire structure is surrounded by another tissue called epimysium, right? It looks very complicated, right? The small piece of meat what you're eating contains all this structure. In fact, these muscle fibers are made up of two kinds of protein, actins and myosins, and they are actually very structured networks of proteins, and that at the end of the day, together everything gives you that fibrous nature when you eat meat. Meat is fibrous. That's something we really want. Not all meat. I'm talking about chicken meat, not fish meat. Okay. They're very, very fibrous in nature. And that is because of muscle fibers. Am I clear? Now, we want to recreate this structure. Remember, we want to recreate this structure to get the fibrous structure. One thing I'm telling you that it's not easy. It's not one type of structure. Muscle fiber is not just like a single fiber. It's association of multiple things. There's a lot of connective tissues are involved here. That gives you the juiciness. There's some fat molecules are also associated inside. That gives you some other taste. So it is a complex thing. Make sense? This is the structure of muscle fiber. Now, let's take a little bit macro structure of muscle. This is microstructure. You can only observe it in, let's say, an ultra microscope, right, or in some electron microscope. But if you look into the macro structure that you see in your naked eye, right, they call it as lean meat. Some people call it as red meat, right? Different types of meat are available. When you cut, let's say, a pork piece, you can see multiple layers, right? A red layer is supported by a white layer or colorless layer. Basically, you have a meat enriched with red blood cells. I mean, what we call myoglobin, which is red color in color, right? And we also have a meat which is not that red in color. And then we have in between fat layers also. So it's very complicated macro structure also. This individual thing inside is made up of this one, right? It's not just this one. The whole structure is made up of several layers, several interfaces containing different types of muscle, right? For example, white portion contains more 75% glycolytic fibers and 17% of muscle fat, right? And red portion contains high only 6.6 about fat and 23% of glycolytic fibers and lot of let's say myoglobin, the red color that is giving. So basically the macro structure of muscle is also very complex. Make sense? So we need to recreate this structure somehow from this one to this one. Now I'm showing the degree of complexity for you. Now this is the structure of, let's say, fibers in chicken or like in pork or in beef. If you could take fish as an example, fish has a different texture. Do you agree? Fish texture is very different than those who eaten fish, they understand, than the chicken texture. And there the muscle tissues are associated in a very different way. So when you want to recreate muscle of a fish, you need to think in differently because their structure, their association is different than the other meat. If you want to recreate, let's say, shellfish, if you want to recreate, for example, prawn or shrimp has a very different structure. Do you agree? It's different than the fish. It's more hard and it is having a very different fibrous nature. So we need to think lot of things here to get that structure similar to the real meat. Lot of things are associated. We need to get this structure. This is my petri dish, for example. I'll take one cell to begin with, and I need to get this one. Of course, I'll supply all the media I need, all the nutrients I will supply. But from one single cell, I need to get the meat. A meat is not one type of cell. The meat is of different types of cells. Meat is not one type of cell. Meat is different types of cells. We have fat producing cells. We have protein producing cells, fibers, right? We have some other kind of cells, adipose tissues, right? We have fibroblasts, skin producing type of tissue. Many things we need. So how do we get from where to start, which particular cell to choose to recreate this meaty structure? And that again requires little bit knowledge of cell biology. Okay, I'll just briefly discuss the cell biology things to choose which particular types of cell we can choose to recreate a meaty structure. Remember, meat is not one type of cell. Okay, there is a class of cells called totipotent cell or totipotent cells. Have you heard about this one in biochemistry? Okay, cell biology or molecular biology, biochemistry, they always teach this one. Even in botany lectures, they teach totipotentin. There are certain cells, they have a capacity to divide and proliferate and differentiate to become an organism at the end of the day, right? There are some cells, they have the capacity to divide, differentiate to become an entire system, right? Can you give me an example? Zygotes, right?
Yeah.
The fertilized cells, right? Egg and sperm, when they fertilizes, we can recreate a human being, we can recreate an pig, we can recreate an animal, right? So they are totipotent. That means those individual cells have the capacity to become a system, complete system, composed of eyes, heart, lungs, and everything. And those are called totipotent cell. I'll come back to your point. Totipotent cell. Is it clear? And usually, not all the animal cells are not totipotent, right? Zygotes are totipotent, but there are some plant cells, they are totipotent. That means you can take one plant, one set of plant cells, you can create the entire plant out of them, including leaf, shoot, root, bark, everything. If you study biotechnology, they teach this in the lab, what we call plant tissue culture. Have you heard about it? Plant tissue culture, they talk about all these things. There are some plant cells, they have the capacity to become totipotent. That means from one cell you can produce the entire plant. Animals, no, you can't take my skin cell, you can't produce Mahesha back, right? It's not possible. But does that make sense? So animals, there's only one, like zygotes, the fertilized egg, they are totipotent, not everything. Then we have a pluripotent cells. Pluripotent cell is nothing but they can differentiate into some types of body. That means, for example, just to give an example, they not necessarily form everything, but most of the things can be produced from pluripotent, just to keep it in a very, very simple terms. Okay? And then we have a multipotent cells. Basically, pluripotent cell is also multipotent cells. They can differentiate and form multiple types of tissues. For example, in this case, you take a heart as an example. Heart contains cells rich in protein, maybe some cells rich in fat, right? And to create, for example, you're talking about liver, right? We need to find out a pluripotent cell that can become an organ, because an organ contains multiple types of cell. Maybe there is a nerve cells are present, maybe there is a fat cells are present, maybe there is a protein rich fibers are present. So they are multipotent cells. From one cell you can produce different types of cells. That's called multipotent cell. Then we have a nullipotent or unipotent. Nullipotent is different. Then I think there's a problem with my animation here. We have a unipotent cells. That means when they divide, they'll become only that type of cell. They will not become another type of cell. Does it make sense? They are unipotent. Now, the totipotent cells are those, they can become an organism. Pluripotent cells, they can become organism, but some limitations are there. Then you have multipotent cells, they divide and differentiate, can become different types of tissues. Then you have a unipotent cell, when they divide, they'll become the same thing. Different thing is not possible. Now, I have to take a cell and I want to get meat. Which type of cell would you choose?
If we need suppose specific meat like lean muscle, then we need some specific cells.
Yeah.
So unipotent maybe, but pluripotent in muscle there are number of these cells. Pluripotent maybe because in this we can get organ. So pluripotent may be another.
May require. Yes, makes sense. So any other answer? Any other answer? Would you choose? He's telling that you are welcome to use unipotent cells. If you just want a protein rich matrix, I don't care about other things. I just want protein fibers now. Maybe I'll go with unipotent cell. And he's also telling that I would also go for pluripotent cell, because sometimes I need protein, a small amount of fat, and it's not just one type of protein containing structure. I need different types of protein containing structures. I need these fibers, I need these connective tissues, I need fibroblasts, I need some extra flavor. Then I was thinking I need to have a cell that is capable of producing all these things. Then I would go for pluripotent cell. You're welcome to go for totipotent cell. It has its own advantages and disadvantages. You need to find a fertilized egg to get those everything that you need. Maybe inside petri dish, it's maybe difficult to have an entire organism. Then you think the complexity. Here I have given a right answer. Depending on what do we need at the end of the day, choose the right cell type to start with, and that is called seed cell, and they belongs to stem cell family, right? Seed cell. Make sense? Is it clear? We need not use all type of cells. We can use a particular type of cells to create a meat structure depending on the end product that we need. I'm going to talk about which one people often use in the next coming slides. Is it clear? Now, those are the stem cell you're talking about. You mentioned about the stem cells. These are the seed cells are capable of producing different types of cells. When we say meat, which type of things we actually need at the end of the day? This is my seed cell. Imagine this is my seed. Which type of cells we do need at the end of the day? One is important thing is that we need muscle cell because that contains myosin and microfibrils and all these things are present there. So we really need it, fundamental. Anything else we need?
For fat cell. Fat cell is also required for marbling.
Yes, exactly. The fat cell is important because meat is not just proteins, it's fat. So fat cells are extremely important because that also gives you the structure and juiciness. Anything else?
The bone cells, these blood cells.
Blood cells perhaps important because some people like to eat meat that is red meat. Maybe we need red cells containing cells. Anything else? Maybe we need epithelial cells because they are skin type of cells. So we might need that. Some people love chicken with skin. Some people love chicken without skin, right? And maybe that we need. Then, do we need sex cells? Might not be necessary, right? They are very minor portion of a given body, not rich in proteins, and it's not actually food. We don't need that one. So immune cells do we need? Immune cells might not be necessary on a nutritional point of view. I have no idea whether that is important for them, the animal cells to grow in the petri dish. It might be necessary. I have no idea. On a nutritional point of view, we don't need immune cells. Do you agree? Nervous cells? We don't want when they're growing, when you touch the meat, it should not become like this, right? Right? It just grow. It should not give any actions for us. It should not scare us, right? So we might not need a nervous cells because it's also a small portion of a given meat. So we might not need nervous cell. Now, is it clear? We actually need, coming back to her question, if she mentioned that we might need a totipotent cell, we might not need a totipotent cell because we don't need everything in most cases. Maybe pluripotent cell is sufficient because it can differentiate into multiple things, not necessarily everything. Am I clear? Or multipotent cell is also enough. Or you can say unipotent cell. I just want muscle, nothing else. You go for unipotent cell. Depending on the need, you select the cells type you need. An important characteristics of this cell type is that the first thing is they should be immortal. We are not immortal, right? We die. Our cells die. But when we die, our cells die because of the proteolytic activity inside the cell. They kicks in, all the life is gone, cells are dead. But when we are alive, if we take our tissue, they are living tissue, right? And one cell become 100 cells, and though from 100 cells you can pick one more cell, it can become 100 cells. From those 100 cells you can do one more 100 cells. So this will be maintained unlimited number of times. You can do unlimited number times of subculturing of the cells. That's the beauty of certain cells. Some animal cells will die after 100 cycles, 200 cycles, 300 cycles, 3000 cycles. But some might not die. They may be forever. For example, if I'm not wrong, cancerous cells, they can divide and go on. But it's important to have cells which is immortal. At least we need to reculture them several hundred times. That's the important thing to have a property. The second thing is proliferative ability, means they have to divide and differentiate. become new thing. And the third thing is that they should be independent of serum. I'll get back to you that later. Serum is a nutrient that we're adding to the media to cells to proliferate, but it's nice to have a serum free media, because serum is expensive and it's not very ethical. I'll get back to that later. So these are the characteristics we need. It's not just creating a bunch of cells. We need to create a structure, because structure produces texture. Food is not just cells. Food is not just proteins. Food is a structure. Food is a texture. And then we have another degree of complexity. Food is a very dimensional system, right? If you take a chicken as an example, take a drumstick as an example. It starts with like this, right? Just take a drumstick as an example. This is the drumstick. Okay. It has a shape, right? My sister's son always likes drumstick compared to the other piece of the chicken. It's his love. He likes to hold it in the bottom and then he wants to tear it off. My sister's another son likes a small nuggets, chicken nuggets. It's his preference. So we human beings want not just proteins. We need the shape. That is very important. That's unfortunate, not unfortunate thing, but we are built like that one. So it's very complex 3D structure we need to recreate protein, just creating a piece of muscle, right? Think about the complexity. But it's very important to know that it's not always necessary that we need that one. Okay, we don't have to have always this very complex 3D structure. Just to give this nice example from GFI. GFI is an institute, extremely nice institute to get more information about alternatives. I always recommend you guys to go and visit this website. They have all the beautiful information you need on alternatives. Okay, GFI. Okay, what I'm telling is that it's not always necessary to get these complex structures, right? It's not always necessary. Just to give one example, you take hot dog as an example. Hot dog has this meaty thing. It does not have a structure. It has a structure, but not complicated like this one. Is it complicated like this one? It's just a meat, muscle tissues are scrambled together and pushed through a kind of orifice, and then you get a hot dog, right? Maybe they are filled in intestinal tissue or some other things, but you will get a hot dog, but it does not have a structure in according to me. The only structure is like this one, that's it. And it does not have any definite structure inside that meat. It just mashed things together. Do you agree? We don't need the structure here, or a complicated structure. Then we can go a little bit higher degree of complication. Take minced beef. Then you need that minced layers of muscle tissues we need, right? Little bit complicated, but not as difficult as a leg piece or a drumstick, right? Then we go a little bit more complicated, what we call steak fajitas, and you have a little bit more structure now, right? This is very soft. Do you agree? And this is not that soft. This is little bit hard and it is more fibrous now. Then you need another level of structure. Another level of structure, texture you need to develop. Now we go to the real meat, let's say steak. Often people use in Americas and Europe. We don't eat that much steaks in India. So we eat steaks. It's very, very complicated, right? You know the beauty of shape? And there is a red layer. There is a white layer, can you see? Right? It's more hard, right? It's more juicy and it has a definite structure and people actually seek for it. A high quality steak should contain all that they think they need. Juiciness, fibrous nature, color, texture, flavor, everything we need. Is more complicated. So the degree of complication is increasing in this direction. And now, depending on the muscle type cells we use, we can create different types of structure. Now going back to Asta's question, do we need a 3D printing? We might need a 3D printing to get this shape at the end of the day. We might not need 3D printing just to create a hot dog. We just need a bunch of cells. Make sense? So depending on the things we need, the complexity increases. Now I'm going to talk about how exactly these things are produced today. Is it boring? Is it boring? No. Okay, now you will explain me. Okay. So I have a petri dish. I will take one type of cell and I will keep it here. Right. Now you will explain me. At the end of the day, I need a stick. This is my stick. Okay. At the end of the day, I need a stick. Okay. What are the processing I would do? Or how do I create that stick structure? Get a cell from somewhere. That's the first step. And he is telling that get a proper cell that is free of something, that is devoid of something, but this has some properties that we need. Okay, those cell types I've already mentioned. So get a cell. Now you guys are done. What is the second step? So I got a cell, I put it on the petri dish. What will be the second step? The second step is that this is the first step, and the second step is that we put the cell here. Somehow we need to make it into more number of cell, and that's called culturing. How do we culture it?
By giving nutrient medias.
So second step is that culture it by giving media. Media is nothing but things containing nutrients. Now can somebody explain what kind of nutrients does potentially we need? Just to think about it. What kind of nutrients we need? Swarna Jyoti, do you have an answer for this?
Growth promoting factors like protein, carbohydrates.
Exactly. She's telling that the growth promoting factors like essential things, like we need some proteins, we need some amino acids, right? We need some growth factors. You must have studied in biology, growth factors, hormones, right? Because animals have so many hormones, then only we have some development in our body, right? So we need hormones, we need growth factors, we need amino acids. Anything else? We maintain a proper temperature. So animal cells usually grow at 37 degrees Celsius because humans are 37, body temperature is 37. I'm not saying that every time we use 37, but 37 is kind of a temperature we can use. So we maintain a proper condition, atmosphere, so that the cell can eat the nutrient and grow and proliferate. Okay, that will be the second stage. What will be the third stage?
Sir, if temperature, then can pH also be a factor?
Yeah, of course. The pH is also important. For example, the body pH is how much? Physiological pH. What is the physiological pH?
6 point something.
6.7, right? So I'm not sure whether tissue culture they use 6.7. I don't remember myself. I never I'd never grown cultured meat myself. Just to let you know, I have done tissue culture because I have a bachelor's degree in biotechnology. So we did at least the lab work to grow meat on a plate. At that time, I never thought about, well, I'm going to eat that in one fine day. We wanted to recreate a tissue. That was a laboratory experiment. But now things have changed just in 5-10 years, right? Okay, good. I don't know the pH, but pH is important. Okay, now I have cells. Now they're proliferating, right? They're one becoming 1,000. Next one, then what? What is the third step we need?
As they are proliferating, so we have to give them a kind of dye system or a mold system so that they can grow it in that particular shape only, and the final shape will be
Yeah, what she is telling that now cells are dividing, they're randomly dividing everywhere. Maybe they're becoming a like a sponge, right? That we don't need. And she's telling that we need to give them a direction to grow so they get the shape that we want at the end of the day. So to do that, we need to give a supporting system for them, right? And that is called scaffolds. Very good point. We need to give them a scaffold, but please note that animal cells have a property to grow that they love to attach to something and grow. They don't want to grow flying or floating. They want to attach to something. That's a property of animal cells. So then they want to grow, right? So we need to provide and encourage and adhering substances, and that is called scaffolds. And the scaffold can be shaped so that we get the end shape as well. Okay, but that is the fourth thing, right? And third one is missing. What is happening here? We have given one cell and it's becoming the same type of 1,000 cells. What is the missing step?
If something is missing there, like we need what we talked about, stem cell.
Yeah.
From stem cells, we need to muscle cells.
Yeah.
For the red color, we need RBCs.
Absolutely. So what she's telling that now I'm having only one type of cells, but if you need a muscle tissue, for example, the whole muscle thing, I need to get other types of cells also in that particular matrix, right? Now that is called differentiation, right? Now the cell has to differentiate, and for differentiating, we might change the media, or we add something, or we remove something, and one type of cell is now becoming different types of cells. That's called differentiation. Okay, that's the third step. And during the second and third step, we have the scaffold. Just to tell, I'll tell that later. What's the last step? What is the last step? I need my stick on my plate now. What is the last step? Harvesting. You harvest the cells, harvest the structure, right? That's the last step. But another last step, that's a good point. After harvesting, what we can do? We can do cooking. We can add flavoring agents, right? We can change the shape in a different way depending on what we need. So that is again, we come back to our original thing. What we do with the other type of food products, right?
Basically-
We can still change the structure, nature of the food by adding additives, flavorings, colorings, and so on. Now you guys are clever.
Basically processing as per our need.
Yes, basically you will do the post-processing as per our need. Fantastic word, sentence to make. Is it clear? Now you're already clever. I don't have to teach the next half an hour. You know how to get the lab. You just have to find a cell somewhere, and a petri dish, and some media, and some scaffolds. Oh my God, it's becoming more complicated. I don't get it right easily. It's a little bit complicated, and let's see how exactly we produce. This is something you explained me already. This is something more illustrative and nicer slide. It clearly shows what you just mentioned. Find a cell from an animal you need, or you like, or you want. Then the phase step, you grow them in a big bioreactor, not necessarily in a petri dish, because we are talking about a large scale, right? And then what Asta was telling, give them a shape. They have to bind into something. That's what we call scaffolds. And then what she was telling, based on the necessity, you do the processing. That's the 4 different phases we have to get cultivated or cultured meat. Now, I'm going to explain you one by one these steps. Listen to me carefully. If there's any question, ask, okay? The first phase is that cell isolation. We need to find the cell that is able to divide and differentiate to become the meat biomass, right? How do they select it? There's a lot of procedures. The first one is directly taking from a living organism. You can take a stem cell from cow. There are some bone marrow has some stem cells, right? A fertilized egg you can take from cow to create a stem cell to have a tissue, right? You're welcome to take live animals. It's also expensive procedure because you can't take, if it's just a unipotent cell, what he was telling, what Mohammed was telling. So basically, you just want muscle fiber. I don't care anything. Maybe you can just take a unipotent cell. Maybe it's easy. But I need usually multipotent cell. I need the cell becoming muscle fibers, fat tissues, connective tissues, and so many things. Then I usually need a multipotent cell. We usually find what we call induced pluripotent cell. That means a pluripotent cell we induce by introducing some genes so that it will differentiate into something else, numerous other things. So usually it is taken from live animals, but please note that you don't have to kill the animal. You don't necessarily have to kill the animal. But the problem is if you kill the animal will die, and quickly cells will also die if you don't harvest the cell quickly. If you don't isolate the cells quickly, they will also die because of the proteolytic activities. Usually, if you look about why we are going for animal-based cultured meat, is for animal welfare. It's not a good idea to kill and create, right? So basically, they give anesthesia to the animal and then they take the tissue. But we will also slaughter because only one time they are doing. They can slaughter and they can take. Clear? The age of the animal, the sex of the animal, everything matters. I will not go on to that one. So many things they will, and health of the animal, everything matters. They will always take a healthy cell. If you don't want to slaughter or take from animal, there are some cell banks. Basically, they have stored stem cells before they have taken it from another animal or something. You are welcome to take from the cell bank. Is it clear? Yeah. But once they take the cells, it's not an easy step. Please note that it's not taking the cell in a forceps and putting on a petri dish. It's not that simple, because those stem cells are now covered by so many other things. We don't need so many other things because they are contaminants in our cell now, right? We need to clean them, we need to wash them, we need to differentiate them. There's a lot of steps involved, and that is done in the petri dishes or culture flasks. Am I clear? It's not just picking one and putting on another thing. No, it has a lot of steps to cleaning, washing, and removing the unwanted debris and getting the right cell that you need. And as he mentioned, it's very important to make sure that that is a viable and potent cell. Again, you need to analyze them. And this is something you don't have to read it now, but the cell types we use is these kind of properties are required. And depending on the requirement, we use different types of cells. Usually, embryonic cells can be used. They are the fertilized egg because they are actually multipotent. They can become into different organs, right? And induced pluripotent cells are also used. And for muscles, especially for meat, they use muscle satellite cells. Basically, they are capable of producing more essential muscle cells because they contains the muscle fibers. They're able to produce muscle fibers, and they're also able to take this called fibroadiponic progenitors. They can become fat cells. Adipose, if you really want to have fat in this one, you can take adipose. You're welcome to take fibrous fibroblasts. You're also welcome to take mesenchymal stem cells because they have the beauty to become muscle, fat, bone marrow, and so on. What I want to tell you, the take-home message is that you're welcome to take different types of cell depending on what do you need. Probably the pluripotent cells are most welcome to take simply because they can become a meat that should essentially contain muscle fibers, adipose tissue, which is fat tissue, and fibroblasts. So these are the three major things we need in meat, and depending on that, you can take any type of cell you need. Is it clear? Now we are coming back to the question, we need to produce them in large quantity, and that is called cell proliferation. As I said, in industrial scale, we don't do it on a petri dish. In laboratory, we can do it on a petri dish or a culture flask, but in industrial scale, they are done it in a bioreactor. We are talking about a big bioreactor. Very big bioreactors are available nowadays. We are talking about 20,000 liter size, right? Like a tank on your terrace, right? That's only 1,000 liter. Now talk about 20,000 liter. So huge bioreactors are available. You need to optimize the process, of course, and those can be used to improve the number of cells or increase the number of cells. Now, the important thing here is that the bioreactor, the second most important thing is that nutrients of the media, you already mentioned, they need proteins, they need carbohydrates, they need other things, hormones, they need growth factors. So many things are present. Please note that this media is way more complicated than the media that you use in your microbiology laboratory. This media is way more complicated than the media you are using in the microbiology laboratory because it's not easy to help an animal cell to divide and differentiate. You need a lot of aids for them compared to a bacterial cells are very crazy cells. They just want to live somehow, right? Animal cells just want to die somehow, right? So this is the problem. So we need to help them. You need to help them to proliferate. Now, one of the important media component we can add is called serum. You know, have you heard about serum? There is something called blood. Have you heard about? The red thing in our body is called blood, right? And blood contains apparently two components, serum and plasma. So red contains, okay, red blood cells are in this one. There's something called serum in the blood, and that serum is actually a mixture of nutrients. It contains so many good nutrients that cell can eat them and proliferate. How do we get serum? From blood. Where blood comes from? From living animals. That's the answer I'm expecting. Come on, don't be so dumb. Blood comes from living animal. Now I'm talking about cultured meat. I don't want to kill animal. Am I sustainable now? Am I following the rules? No, right? If I take blood or serum from a living animal, it becomes not comes within the animal welfare stuff. Am I clear? And serum is so expensive, extremely expensive, because usually that is taken from the uterus of a cow. Cow has a small uterus, right? And you can imagine, if I want this much amount of serum, how much blood I can collect from uterus and then serum, which is a very expensive and complicated process to get a small amount of serum. And the whole thing becomes super expensive, and whole thing can become less animal welfare stuff. Is it clear? So serum is not a go. But if you find serum is a fantastic nutrient. Usually serum is also used, unfortunately, but it's nice to have a serum-free media. If you don't add serum, you need to add components present in serum artificially. You need to add hormones, you need to add growth factors, you need to add proteins, you need to add vitamins. Everything should be added separately, one by one. Am I clear? So serum-free media is recommended, and there are certain governments have instructions saying that if you use serum, you can't sell it in my country. Am I clear? There is a government regulations also in certain regions of the world that you can't culture with serum. Make sense? But nutrients are expensive, things are becoming more expensive. Now, once you have supplied the media to cells and they start growing, but as I said, animal cells likes to attach to a surface. You need to give them an anchorage, a body to attach, and that is called scaffolds. And different types of scaffolds are available. For example, the one of the things we have is scaffold is clear. It's basically a thing where the cells will attach and then they proliferate. Okay, it's very important to know that these scaffolds should be biocompatible. Do you agree? Scaffold should be biocompatible. You cannot use toxic scaffolds, because at the end of the day we will make burger out of it, so we can't use it. And they can be porous or they cannot be porous. Porous may be helpful because then the nutrient can pass through these beads and then they can go and attach to the cells, and cells can easily eat the media. Make sense? Is it clear? And porous can be of different shape. What Asta was telling. If you want a particular shape, maybe you can find a particular type of porous with that particular shape, so you can use it. So usually different types of scaffolds are available. 4 types are called microcarriers, porous, fibers, and hydrogels. Microcarriers are very commonly used. They are small beads. Usually they are also porous, right? You can add into the media together with the cells, and the cells will attach and they grow. Okay. Then we have fibers. Basically, fibers are elongated. This one, they have a small holes, or they're also porous, or maybe having a channel, and the cells can grow on top of them. You can get fibrous structure. Okay. Then we have hydrogels. They're special ones. They can absorb more water, the hydrogels. So you can use different types of scaffold. There's a huge research on getting the right type of scaffold. And you were asking about 3D printing. You can use a scaffold which is 3D printed. At the end of the day, you can get a shape you need, but it's much more complicated. Why? Because if you want to get a shape of stick, you can easily make a 3D printed shape. But the problem is the cell has to grow in the same region properly. You need to have meat, muscle fibers, fat cells, and red blood. This is very complicated, but people are working on that one. Okay. Now, if you're coming back to the characteristics of the scaffold, the most important thing is that they are biocompatible. Okay, they are not toxic. The second thing is that it is also possible to eat scaffolds. For example, you have a bioreactor, you added scaffold. This is my scaffold. Okay, cells are growing on the scaffold or in between the scaffold or inside the scaffold, whatever it is, right? Two things can be done. He was telling you have to harvest it. Harvesting is nothing but removing the cells from the bioreactor and also from the scaffolds. But some instances, you don't have to remove them from the scaffolds. You can eat cells and also scaffolds together. You get a structure now. Am I clear? That's the reason why scaffolds can also be an edible component. For example, cellulosis, hemicelluloses, pectin, and those kind of fibrous structure can be used to create scaffold. Now we can go back to our sustainability phenomenon. I extracted proteins from microalgae. A lot of fibers are left. Maybe I can use that fibers to create scaffold here. Nothing is wasted. Am I clear? So we can use natural fibers to create scaffolds. Am I clear? Because scaffolds are also edible. And then important thing is that we need to talk about cell differentiation. That means inside the scaffold now the cells are growing. They're rapidly multiplying, but they're not differentiating. Division is different, differentiation is different. Division is basically 1 becoming 2, 2 becoming 200, that is division. But we need to have a proliferation. That means that now one type of cell should become another type of cells. For example, we need to have some muscle fibers, also adipose. We need to have some fat cells. We also need to have some fibroblasts. So we need to have multiple. Now we need to tell the cells enough dividing. Now you differentiate. Am I clear? They are continuously dividing to same type of cells. If you just let them grow, they just become the same thing, big biomass. We don't want that. We need to become them in different types of cells now. Okay, that we need to induce, and that is called differentiation induction. That can be done in many ways. One of the things I read was that you need to reduce the growth factors, because growth factors help them dividing, and you have to increase the signaling molecules. You need to signal, okay, now it's the time to differentiate. So you can add them. Basically what they do is that they change the media. If you change the media components, they will forget about division, they will start differentiation. Okay, because that new media has new components. Now they realize, oh, enough dividing now, let's differentiate. Am I clear? You can also welcome to use GMO cells. Apparently they add, because they containing some probably a signaling stuff. So you add a small amount to the reactors, and then they will help, they will send signals, okay, now let's differentiate. But if you add GMO cells, you can't sell it in some part of the world. Let's say European Union, there were strict guidelines on GMOs. GMO means genetically modified organisms, where the DNA is changed. They add a foreign DNA. Okay. You can also change scaffolds. Apparently, they can also they are growing on one type of scaffold. Now you change the scaffold to another type. Then they realize, they wake up. Let's differentiate. Am I clear? All right. So now we come back to the last phase of cultured meat. Once you have this bioreactor containing scaffold, and scaffold attached to the meat cells and fat cells and fibers and so on, you have two options. Oh, I am coming back to the same thing what I was telling you, that either you harvest along with the scaffold, or you remove the scaffold and remove the cells, right? To remove the tissues and cells, they use enzymes, thermal mechanical methods. Enzymes, you can use proteases, and thermal, they heat it so that the attachment is removed, and mechanical methods, you can just shake them, then they detach from the scaffold, right? There's a lot of ways to this one. For harvesting, you can sediment the cells, you can centrifuge the cells, you can harvest them. There's a lot of, and you can also do filtration after centrifugation, because you need to remove the cells from the media components. We don't want to eat media, we don't want to drink serum, we just want to eat the cells. Am I clear? So harvesting is also important. And that is not the end of the story. In most cases, you get a meat which is like this, without any structure, shape, and so on. Again, you need to give them shape and structure. Again, you come back to our own plant-based world. You're welcome to use transglutaminase enzyme that can do more cross-linking. You're welcome to extrude these fat and muscle fibers together, right? You're welcome to add some colorants. You're welcome to add some more fat. You're welcome to add umami flavors, aspartic acid and glutamic acid, right? It's not that you get the end product from the bioreactor. No, it's not necessary. It has to be again processed to get the final structure, maybe minimally processed compared to plant proteins. Am I clear? And then you get your product that you need. You are welcome to cook and eat. Don't eat raw. To characterize the structures, we have a lot of analytical techniques. I don't tell that, just to let you know that it's not a simple research. The microstructure and the macrostructure of this meat, cultured meat, can be evaluated by numerous analytical techniques. At the end of the day, after adding additives, after doing some processing, the cultured meat can look like this. Can you see? It's not bad, right? It's not super bad. It looks like a thin slice of meat. You have a red one, we have a lean meat, we have pink meat, right? So it's possible to produce a meaty texture and flavor using cultured meat. And now we need to discuss about what are the challenges associated with cultured meat production, and that will be a group discussion. I talked about how to produce cultured meat. You also mentioned to me, oh, why this is looks so difficult. What could be the potential challenges of production and also eating cultured meat? You will have a group discussion. I will give you 5 minutes to discuss. We discuss and then we take a break. Okay, what did you guys discuss? So they are telling that it's difficult to mimic taste and texture. One point, Asta, one important point.
That's the consumer acceptance, because most of the people who are fond of meat
Yeah.
will like that chewy, fibril
Yeah.
texture with the original taste
Yeah.
and that.
Yeah.
So consumer acceptance might be a big hurdle for this, and relation of the social and ethical issues also
Yeah.
can be there,
Yeah.
because people like the natural things they are getting from the nature.
Yeah.
So it is not something which is from the nature and we are creating in the lab.
Yeah.
So there can be social and ethical implication with
Yeah.
the consumer acceptance on the basis of that particular texture, desirable things.
Yeah.
As we have different types of slaughtering methods,
Yeah.
that is jhatka, halal, and
Yeah.
Jewish method.
Yeah.
So it is not basically coming from that slaughtering method.
No.
Right?
So
So-
exactly, yeah, because we have a particular group of people, they would love to have the animal that has been slaughtered by the same method according to their religioustic
Yes.
purposes. Would that be met? That's a very good point, right?
And another things there is no specific regulations or guidelines.
There is no
Like-
specific regulations and guidelines. Okay, I'll get back to that point. Yeah.
Conditions are very specific. In
Yeah,
terms of that
condition.
pH temperature, if it changes for certain reason, then the whole cost will be go in toss.
Absolutely. We are talking about 20,000 liter bioreactor, and I will hire this gentleman to maintain the bioreactor. And that gentleman is sick today, and he forgot to press a button. Things might go change, right? It's very difficult to maintain. I agree. This is a process thing. You guys are engineers. You know better than me how things can go wrong in large scale, right? Yeah. Yeah.
So, and the equipments which we use are very complex and hard to operate. So we need training.
Yeah. We need trained personnel to operate the big equipment, but that also creates technical jobs. Just to let you know, it's not necessarily a negative thing. It could be a positive thing, because we think science in 2 different ways. Like, we need highly skilled people, we need semi-skilled people, we also need unskilled people, because that's a different job market. Let's discuss what you guys discussed. Yeah.
Sir, for production, it will be very expensive.
Yeah.
Like serum, it will be very expensive.
Cost is always depending on demand and consumption and production. They're all interrelated. Things become cheaper if more and more people buy that one, because they can find alternative solution to make things cheaper. Good point. Any other thing?
As the production is costly, then the consumer means the product will also be costly.
Yes, yes,
So-
exactly. So we discussed that point. So ingredients are expensive, final products are also expensive. Yeah. Any other thing?
And sir, the environmental impact, the energy use.
Yeah, the energy and environmental impact. Very good point. Environmental impact plus energy. Is it matching? That's very important to know because at the end goal is nothing but animal welfare plus environment.
Culture meat should be differentiated means and negative impact of that meat.
Yeah, you mean during the differentiation
Yeah.
process. So maybe, yeah, it's important that we get the right type of stem cell. They differentiate into right type of tissues at the end of the day. If they differentiate into something else, we might have some problem. How do we control the differentiation so that they produce right type plus right amount of cells? We need more muscle fibers. We need less fat. We don't have too much fat and less muscle fibers, right? We need to control differentiation, and that requires optimization. Completely agree with you. Good point. Very good point.
It requires constant temperature, oxygen, and nutrients,
Yeah,
which can lead to significant energy consumption.
absolutely. So, yeah.
And the cells that we are using for the cultured meat, we don't know when the cells will get mutate
Oh, yeah. Yeah.
or get ruptured. So that also
Exactly, because we want to use the healthy stem cell. They're healthy enough to grow healthy meat. We have no clue what else would happen if you continuously regrow them, reculture them again and again. I know they are immortal. You can grow from one cell to another continuously, what we call subculturing, reculturing. But at some point, something might go wrong. You never know what would happen. Good point. Yeah.
We have a lot of steps to optimize.
Yes.
So, for which we need food technologists as well as biotechnologists.
Yeah.
So there is a huge scope for research in this field now.
Absolutely. There is a huge scope for R&D because if you look into this diagram, you see how many expertise we need, what kind of experts we need.
Exactly.
We need cell biologists to find out right type of cell, right?
Yes.
We need tissue engineers to proliferate them. We need media guys, not those newspaper media guys. We need media guys to produce serum-free stuff. We need media guys to produce proteins. We are losing the screen. We need people to produce same pH. We need somebody to maintain the bioreactors, right? We need process engineers.
Yes.
We need technologists. We need electricians.
Yes.
Right? We need somebody to make a stainless steel metal that is not releasing corrosive substances. We need scaffold engineers, hydrogel makers. We need the final burger makers also. We need chef, right?
Yeah, and one of the very crucial thing is we need to, after setup of all this, we need to establish with different sort of cleaning and other sort of sanitation practices. Because
yeah.
these are very different from the traditional ones.
Absolutely. For after slaughtering the animal, maybe they clean it in a different way. Now they are being grown in bioreactor that contains pipelines, tubings, and so
CIP and all.
Yeah. I'm not an engineer though, but you imagine how much thing we have to think about cleaning because even the cleaning the UHT processing system, milk system, it's not an easy thing because you have biofilm formation,
Yes,
blah blah, so many other problems, and how do we maintain that one? So yeah, good point.
So just to add on to that, like even we have established systems for dairy like cleaning and all.
Yeah.
It's called CIP clean in place.
Yeah.
So apart from like normal hot water, alkali and acid cleaning,
Yeah.
which every dairy industry does on their regular basis.
Absolutely. Yeah.
So even after like concentrated like acid cleaning also,
Yeah.
they in a once in a month or maybe
Yeah.
we actually need even Tetra Pak systems also. We need to manually open all the plated areas and then clean them. I've personally seen them scrubbing with brushes
Absolutely.
and then again doing the CIP so as to remove those residues.
So the biofilm is the biggest challenge.
Challenge, sir.
So it's not because the contamination, what you mentioned,
Yeah.
you forgot to mention about the bacterial contamination. Basically, these cultures can also be invaded by bacteria because you're supplying numerous ingredients. Bacteria want to love, and the bacteria are crazy. They love acid, they love base, they like high temperature, they love low temperature. They're crazy, they're ubiquitous, they can grow everywhere, right? And then we have to clean them.
Yes. So we have different sort of residues like biofilm, biological residues,
Yes.
inorganic, organic, all sort of residues, all sort of
Exactly. The
contact-
media component may be stuck in the joints,
yes.
and now they are building there, and they're becoming toxic.
Yes.
I can't, the acid cannot penetrate to clean them. When I open up the nuts and bolts to clean and
Clean.
scrub them,
Yes.
how much expenses they, right? Look about the steps, how many things we need to solve here. They're all expensive. That adds to the cost of the final burger you're eating. Is somebody having a good feeling about cultured meat? Please talk about that.
First of all, animals will be saved. There will be no slaughtering.
Can you hear that one?
Yeah.
There is some good thing. One, no slaughtering. Animals are saved. Animal welfare is there. Very important point.
Second, production, we can, if we optimize all the processes, then we can produce as per our demand. And
So mass production is possible provided you optimize everything. Bioreactors can be big size nowadays. We're talking about liters of liters bioreactors can be made, right? It's scalable. Less space is required because animal husbandry requires a lot of space, a lot of water, a lot of things, but here less space. Yeah.
And one more thing, it will be contamination free. As in case of slaughtering, there are a lot of chances of during handling, contamination chances are more high.
Exactly.
So if
Food
we
poisoning from meat is biggest challenge throughout the world, including India. Meat is spoiled a lot if they forget to slaughter properly. Immediately after slaughtering, they forget to store properly. You will have a serious food contamination. Foodborne illness is one of the biggest challenges, even for death in developing and underdeveloped countries, including our own country. Right? So that's a big challenge.
One more thing, we can craft food as per our demand. Some in case of meat,
And-
saturated fats are high. People do not like, some who are concerned with health, they do not depend on it. So we can craft food as per our research.
Very beautiful point. We can craft food as per our desire, right? I don't want to have more cholesterol. I don't want to have more triglyceride. Maybe we can craft the food enriched with protein, less unwanted fat. If you're really interested in reading more about culture, I really recommend this review article, which is published in Advanced Sciences. I really enjoyed reading it. So just go through this article. And there is also a very nice article written by GFI, Good Food Institute, the US one. They have a fantastic article about cultured meat and problems, regulatory guidelines, and so on. So you're welcome to read through these two articles. Is it okay? I only covered the fundamentals. I did not go very deep into the subject, but you are welcome to refer to these two articles. Now I'll move on to the other two topics of today, microalgae or algae and insects. As I mentioned, I have covered these two subjects in detail on day one. So I will not explain them again in detail. I'll just skim through the slides. Is it okay? That is because at the interest of time, plus I've already covered them. Today's main part was cultured meat and I have taken enough time on that. Is it okay? So algae are nothing but they are algae. They are photosynthetic. They are aquatic. They love growing on water. The beauty of microalgae is that they can produce food by using water, sunlight, and some nutrients. Right? They are very sustainable. That's the reason. They produce food. They're photosynthetic because they are not dependent on someone else to produce food. They produce food themselves that by using sunlight, water, and some nutrients. And that is very advantageous for us because we don't have to rely on animals. We don't have to rely on even on plants to produce, let's say, proteins, let's say fat, let's say carbohydrates, let's say vitamins, let's say polyunsaturated fatty acids. Everything can be produced by this beautiful organism. Sometimes they grow fast and sometimes they grow slow. They can be grown in open pond. They can be grown in bioreactors. Microalgae are very beautiful because they can also grow in the absence of light. They are called heterotrophic. They can also grow in the presence of light, also in the absence of light, because in the morning they become photosynthetic, in the night they become heterotrophic. We can say that mixotrophic mode of growth. They are very versatile. Are you understanding what I mean? They are very versatile because if they have light, they will grow. If they don't have light, they will grow. Light and no light, still they will grow. They need some amount of nutrients because, again, the world works on mass balance. You can't create a new mass without adding feed compound. That's important. So microalgae also need that. They get the energy from the light, they get carbon from carbon dioxide, they get hydrogen from water, so they can create new proteins and so on. I've already covered all these things. Seaweeds are also similar. The only difference between microalgae and seaweeds are macroscopic, they're bigger. But usually microalgae produces more proteins than seaweeds. So seaweeds are not generally used for protein purposes. They're mainly used for carbohydrate purposes. For example, sulfated polysaccharides. All this can be produced from seaweeds. As I said, microalgae contains proteins, polysaccharides, lipids, pigments, vitamins. Basically, they are mixed of everything. The beauty here is that you can train them so that they produce more proteins, or you can train them so that they produce more fat. You can train them so that they produce more vitamins. That's the beauty. You can train their metabolism. They're very flexible in terms of their metabolism. Depending on the type of the strain, depending on the conditions you're providing to grow, they start producing one or more molecules in large quantity. For example, Chlorella vulgaris, it's a well-known microalgae used for production. They can produce more protein, up to 50% in some situation. For example, if you supply more nitrogen, they produce more protein. In some situation, you stress them, they start producing more fat. In some situation, they produce more colorants, for example, carotenoids. You take Haematococcus pluvialis, that is a microalgae that produces astaxanthin, a red color pigment. So that is species specific. It's known to produce astaxanthin. So what I wanted to say that you have a lot of options in microalgae. It's not just one microalgae. They say, I don't know, maybe 100,000 species of something. So many species are available. They're unique in one or other way. But we have not exploited everything. Chlorella vulgaris, Haematococcus pluvialis, Nanochloropsis, Senedesmus. These are some of the well-known microalgae we are training today. And you must have heard about spirulina, Anthospira platensis or platensis, whatever that is spirulina. It's not microalgae, it's a cyanobacteria, but they are same but different. One is prokaryotic, this is eukaryotic. Botanically, they are different, but on a productivity wise, they are similar in the sense Anthospira produces a lot of proteins. A lot means up to 90%. So this is just a graph I wanted to show that the protein content when you compare animal-based versus microalgae, they are a lot. Basically, if you want to say about the weight basis, microalgae are really abundant in proteins. I also mentioned this thing earlier in terms of protein quality, they are not that bad. The protein quality is equivalent to plant and animal sources. For example, if you look into spirulina, they are really good in terms of their nutritional quality, very comparable to egg or soybean protein isolate. Okay. And I wanted to mention one simple thing is that it's already in the markets. There are some industries or there are some countries have approved that you can market them. They're available in capsule form, they're available in powder form. But one of the challenges here is that the good thing is they contain everything that we need, healthy. Can you talk about challenges? Toxic, and they come up with something else. That is this is one of the challenges, very similar to cultured meat. Yeah. The color. They always come up with this green color because of chlorophylls, and we don't want to have color, right? When you have a green color, if the food becomes green, first problem. The second problem is green color absorbs light. it will become a photosensitizer. Come back to my previous lecture, what photosensitizer can do. They go to higher energy state, they can do all the radical and non-radical mediated reactions. They can oxidize not only the protein which is coming from microalgae, other thing you have added to food, that will also be modified. So that is one of the challenges. So you compromise the product quality. Somehow we need to remove the color. How do we remove the chlorophyll? Extraction, purification, another problem. Otherwise, you can go GMO approach, genetically modified organisms. You knock out the chlorophyll gene, you can produce. But again, a problem. GMOs are not allowed to eat in some countries. They have their own advantages, and of course, we have non-GMO way to produce also. That's another story. I don't teach all these things today. Just to let you know, they have its own advantages and its own disadvantages. The biggest disadvantage, according to me, we can't eat the whole microalgal biomass.
What's the to do?
We can use Anthospira, spirulina, because they have a very weak cell wall. But if you want to eat Chlorella vulgaris directly, remember my diagram? Protein is inside the cell wall. Cell wall of microalgae are very, very strong. And if you eat them, the cell wall has to be broken down in your stomach. To do that, you need cellulase enzyme. Unfortunately, human beings lack cellulases, hemicellulases, pectinases. We don't have it. So actually the protein may not be digestible, may not be bioavailable, and that is a challenge we have to solve here. Is it clear? I don't go deep into that because I've already explained that one. Beauty is that you can grow them in large quantity. This is the Hematococcus pluvialis, what I was talking about. It produces a beautiful red pigment. We can use it as a colorant to color meat. Now you can see that how two industries can club. Right? Biggest challenge I have already mentioned, and also today they are very expensive. They may be up to 10-fold more expensive than the plant proteins, simply because it looks all fancy and nice. I say, oh, less land, less water, more product, high protein, but they all need cultivation strategies, big capital investment. You can't grow microalgae in Copenhagen, in Denmark, easily. Why? Because
Sunlight.
we lack sunlight. We can't make open fold. And microalgae for growth, they also need some temperature. They love heat. Copenhagen temperature minus some degree Celsius during winter. We can't do it photobioreactor outside. We need to maintain the temperature inside. Maintaining temperature is so expensive. Am I clear? So these are the problems we are facing. Any question about microalgae? It has a lot of potential, but it also comes with its own problem. I have already taught you this one, so I don't go into detail. Then insects, we discussed a lot about insects. We discussed somebody who have a startup. What was the name of the startup? I forgot. Hell Locust. Hell Locust startup, they will be there. We're going to discuss more about that one. Fantastic source of proteins, high quality proteins, in fact. In fact, they also come up with very good minerals, very good amount of PUFAs, polyunsaturated fatty acids, so win-win benefit. But the biggest challenge here is that consumer acceptability. Many people think they are bugs. Many people think they carry diseases. So consumer acceptability is really, really low for insect proteins or whole insects. However, we discussed about the strategies. Just to mention that they are very sustainable. With less amount of water, less amount of land, you produce more insects. Plus, when you see the biomass, 80% of their body can be edible. Only 40% of body is edible in case of cow. The rest is bone, hair, skin, and other thing. But here is more edible. So it's also sustainable, high quality protein, very digestible. But the problem is consumable acceptability. But to avoid that one, we can convert them into ingredients, right? You don't have to eat whole insect, convert them into ingredients, then they can be converted into a product. Extrusion, fermentation, 3D printing, Any different technology you convert protein into product. So we need to take plan B. Plan A is directly eating, not working. Go for plan B. Usually, insect protein flour is used in bread making today. It's not universally adapted. I can only see many articles on bread making. That means they take X percentage of wheat flour and replace it with Y percentage of insect flour, and then they get high quality nutritious bread, because bread may have a very low protein content, very low amount of good amino acids. Then you add a portion of, you can't make bread completely out of insect flour because of the gluten problem. You need that networking, but you can take a portion out of them and add this portion. You can get a high quality bread. That's being done a lot. The biggest challenge is the consumer acceptability. I read a recent article published. They clearly say that it's going down. It's not going up, because many consumers still think that insects are no-go. Cultural problem. Okay? That's the end of my lecture number 15.